What's the difference between a tomato and a fish? Everything appearance-wise. But it's what's on the inside that matters. Where am I going with this? About 4 years ago, a company called DNA Plant Technology of Oakland, California, attempted to insert a fish gene into a tomato to increase its frost resistance. This would allow tomatoes to grow in colder climate and create another transgenic plant. However, the project showed little promise and received a highly negative response from consumers, so it was not pursued. In case you missed it the first time, I said the scenario would create another transgenic plant. Let's hear that again. Another transgenic plant. So, what is a transgenic plant? Trans means into another, and gene is the unit of hereditary that can be passed on. So a transgenic plant is a plant that has a gene incorporated into its DNA that can be passed on. The first transgenic plant was made in the early 1980s, and it was on a tobacco plant that expressed both antibiotic and herbicide resistance. So yay, tobacco. Excuse me, how do you make a transgenic plant? For curiosity reasons. Well, it all started when a group of researchers made their way into the forest. Nothing but the sounds of nature encompassing them. All of a sudden, as if something reached out and tripped one of the researchers till they were face first into the earthy floor. The rest of the group saw what had done the cruel deed. It was a gall, is a kind of swelling or growth on the external tissue of plants. They are a reaction caused by a bacterial infection from Agrobacterium, which is common soil bacteria. The bacteria makes its way into the plant through damaged tissues and transfers a gene that makes the plant cells produce the nutrients that the bacteria needs. This happens as the tDNA of a bacteria DNA molecule, or more specifically in the TI plasmid or tumor-inducing plasmid, is incorporated into the plant genome, which causes cells to multiply intensely to produce the needed nutrients. What was discovered was that the bacteria can be forced or modified to integrate completely different and wanted genes into the plant genome. By including desired gene sequences between the border sequences of the tDNA section, the TI plasmid is ready to be used. The modified plasmid is introduced into bacterial cells, and eventually the wanted gene is incorporated into the genome of the plant. Agrobacterium with the modified plasmid is integrated into damaged plant tissue, and the transformation of plant cells occurs. By changing the plant nutrient composition, scientists can then make the cells containing the foreign gene start to divide and change the shoots, which is the visible part of the plant. Shoots are then transferred into another medium that enables the formation of roots. With one genetically modified cell, an entire transgenic plant can grow. But like everything in life, it has its pros and cons. While it can increase yields and reduce the use of pesticides and insecticides and become virus resistant, it's also costly and can become ineffective over time. It also disrupts biodiversity by maybe favoring an organism. And not to be a Debbie Downer, but there are also social and ethical implications to this relatively new biotechnology. For example, who will have access to this technology? Because developing countries could benefit from it, but will most likely be affected by deployment and not have access to the product. Will it affect human and ecosystem health over time? Also, what are, if there are any, the consequences to blurring the species boundaries? Thankfully, in Canada, safety assessments are mandatory for each transgenic plant. Scientists review the microbiological and chemical safety of the plant and its potential to cause allergic reactions, its potential of introducing new toxins, how the plant was developed, including any genetic changes made to the plant, how the genetically modified plant compares to an equivalent non-modified plant in terms of nutrition quality and what it's made of, for example, the fats, the proteins, and the carbohydrates. These are principles that Canada has come up with based on the opinions of international experts such as the World Health Organization, the Food and Agriculture Organization of the United Nations, and the OECD. Says a 17-year-old! I know. But this biotechnology is not far off from what I'm learning today. To make the TI plasmid, ligase is used, which is also used in DNA replication. And that means proteins and enzymes need to be synthesized. The bacteria helps to regulate the gene and incorporate it into the plant's DNA. For the transgenic plant to grow, DNA replication has to occur, as well as photosynthesis and cellular respiration. The biology I'm currently learning is the foundation of this biotechnology. And maybe next time they decide to put an aquatic gene into a tomato, they should give it the ability to float. Rubber duckies are getting old.